Good evening. Now we are to discuss the Y-Str analysis. It is a processor method which is as a novel tool for fetal sex determination. Now, what is fetal sex? As you know, the male baby, female baby, which are exactly determined by different processes from the traditional concept, and also different scientific methods are there. Exactly, the fetal sex is determined after 7th week of gestation, but it is exactly ineffective before 5th week of gestation. The embryology concept, possibly you know, the reproductive system or the genital system of fetus develops from a common genital duct. Possibly you have the common embryological concept. Such genital duct by different protein factors, hormones, genes, which is exactly divided into two parts like the Mullerian duct and another is Wolffian duct. Mullerian duct, in another way, it is also called the paramesonephric duct. The Wolffian duct, also called the mesonephric duct. Now, the paramesonephric duct, after so many conversion and changes, it is finally develops the female genital system, and the mesonephric duct or the Wolffian duct finally develops the male genital system. Now, the different types of methods for the sex differentiation in fetal. Now, we are to discuss the very latest method, which is exactly discovered in 2011 by different scientists. After that, modified the method, but first it was studied by Diana W. in 2011 August 9. After that, it was modified by Biani et al. 2015. Before that, it was also modified 2014 by other scientists. Now, it is the latest non-invasive method for detection of the fetal sex, which is exactly the method, the Y chromosome process, which is called the Y-Str process. Now, why the Y chromosome is important in sex differentiation? Possibly, from your earlier concept, you know the XY chromosomal process. From the karyotyping, from the chromosomal concept, we know the Y chromosome is responsible for the male child, and X chromosome is responsible for the female child. Though we know YX and XX. Now, in Y chromosome, there is the SRY gene, possibly you know. This particular gene is very responsible for the sex differentiation. It is located in the Y chromosome at P11.3 locus. Possibly you know the P, the petit part, the short arm of the chromosome, and its locus is 11.3. From this locus, from this gene, different types of protein factors are synthesized, which are responsible for the development of male child. The SRY codes for a regulator protein, which is responsible for this differentiation. There are so many proteins. Now, we are coming next for the discussion of such different proteins and their role in differentiation. The SRY gene regresses paramesonephric duct or predominates mesonephric duct, causing male genital system development. Now, we should go to see the embryological timeline for sex differentiation. As different weeks of gestation, the different events of sex differentiation or the reproductive aspects are developed. Like in gestational age in week 1, at that time, inactivation of one egg. At 4th week, development of Wolffian duct. At 6th week, development of Mullerian duct. 7th week, seminiferous tubule differentiation. 8th week, regression of Leydig cell appearance and testosterone synthesis. The regression of the MD and LC appearance starts. The testosterone synthesis starts at 8th week. At 9th week, masculinization starts, regressions of WD means the Wolffian duct, MD means Mullerian duct. 12 to 14 week, development of testis, penis, spermatogonia, etc. At 16 week, development of ovarian follicle and Leydig cells. At 17th week, testosterone synthesis age very high and Leydig cell regression occurs. 20th week, at that time, reduction of testosterone occurs. At 24th week, the rise of ovarian follicle, and 28th week, the rise of oogonia and descent of testes. These are the scenario of different events regarding the reproductive development in male and female fetus. Now, the different methods of sex determination. Number 1, from the traditional concept, different myths and facts, we know different myths and facts. As from our grandparents, we know the different concepts regarding the pregnant woman who will bear the male or female child, like the eating habit, postural habit, the health status, the skin color, and so many other signs and symptoms. They studied exactly from their previous experiences. Though exactly there is also the scientific correlation, but in all cases there are no such scientific relations. Now, the other different methods are also there. The traditional concept, like the baking soda test. Now, what is the baking soda? Possibly you know the baking soda. Exactly used it as the baking powder for production of bakeries. Now, the baking soda test, how is important? Why is it important? Exactly during the pregnancy, the pregnant mother's urine, when we acidic, the baking soda forms the bubbling. But why? Due to rise of pH, the rise of acidic condition. Possibly at that time, the male baby is formed in the mother. Another different types of traditional concepts are also there, like the skin complexion, like the eye colors, and also the skin folding. Such and such different types of traditional concepts are there. The other scientific method, that is the USG, we know, ultrasonography. Different types of parameters have been studied from ultrasonographic studies, like the femur length, the total height, total body surface area, such and such different parameters are studied in ultrasonography, which determines the male and female babies. The pre-implantation genetic diagnosis. The pre-implantation genetic diagnosis, what is this? The genetic diagnosis before pre-implantation, it is exactly the genetic analysis or gene analysis of the mother and father. Another process, the chorionic villus sampling, CVS. From the chorionic villi, possibly you know the chorionic villi in the placenta. From there, by the invasive method, the sample collected from there, from the mother's womb, and are analyzed for detection of male and female babies. Another test, amniocentesis, from the amniotic fluid collected from the mother's womb, and the AFP test. What is AFP test? The alpha fetoprotein test. Possibly you know the alpha fetoprotein. The alpha fetoprotein is very important parameter for the detection of male and female baby. Not only that, AFP also is important for detection of different anomalies, the pathological conditions also, like the tumor, cancers, trisomy, such and such different types of anomalies. The another latest method, that is cell-free fetal DNA testing, one of such sensitive method, which is Y-Str method, which we are just going to discuss in detail. Now, what is Y-Str analysis? Y for Y chromosome. Now, why the Y chromosome is important in this process? Commonly we know the presence of Y chromosome in maternal blood indicates the male baby. Now, why Y chromosome is available in maternal blood? Exactly, during gestation, during pregnancy, the placenta is formed, you know. In the placenta, there is a trophoblastic cell. Such trophoblast cells, which is exactly very fragile type of cells. Why? Due to trophoblastic oxidative stress, such cells are becomes fragile, and the Y chromosome and the cell-free DNA are exposed in the maternal blood. Such DNA and Y chromosomal genetic sequences are analyzed by PCR, as we are coming next, where if we find the STR, short tandem repeat, means the different types of repetition or different locus, that is called the short tandem repeat. It is a molecular method used to compare alleles repeats at specific loci in DNA between two or more samples. It is a microsatellite with repeat units that are 2 to 7 base pairs in length, which can be measured by PCR or the very latest model is the RT-PCR. Now, what is the principle of Y-Str analysis? Exactly, the Y-Str analysis is done from the cell-free fetal DNA from maternal plasma or serum, which is used for molecular diagnosis of prenatal sex discernment. Now, what are the basic steps of Y-Str analysis? Extraction of nuclear DNA from cells or samples from the maternal plasma or blood. Next, the cell-free nuclear DNA, how is separated? Exactly, it is separated or isolated by using a specific type of kit. The kit is QIAAMP DNA mini kit. You see, from the study material, as I said you, with special modifications done in the extracting procedure to concentrate and obtain minute quantities of fetal DNA together with maternal DNA from maternal plasma. After that, certain regions of DNA are extracted and amplified by PCR. The PCR, possibly you know the working principle of PCR, which copies the DNA and amplified. Now, the amplified DNA from the PCR which are after that the repetition of looking is done from the Z electrophoresis. If the repetition of looking is found very much, which determines the male child, as it is found in the Y chromosome. But if there is no any signal for the multiple loci, that indicates the female baby. It is exactly the interpretation. Here you see the 16 to 17 Y-Str loci indicates female fetus. No Y-Str signal indicates female fetus. Exactly this is the main criteria for interpretation of this method for determination of male and female fetus. Now, the importance of this study, another importance. The sex determination is the primary goal of this process. Out of that, in different aspects, in different fields, it is also implicated, like the crime detection of sexual assault by vaginal swab, presence of male and female DNA, individual identification of different individuals, identification of crime from footprint, touch DNA from skin autosomal Y-Str. As you know, different forensic studies are also there. Here I wish to add another importance, like it is also used in determination of different types of trisomy anomalies, like the 13, 21 trisomy, the Down syndrome, such and such different types of anomalies are also determined by this process. Now, not only the advantages are the main target of this process, there are also different types of disadvantages. But these disadvantages are not very important. Exactly, it is time dependent for 5 to 9 days, less effective for degraded DNA, having the low sensitivity for other DNA. Loci are not independent in Y chromosome as a random matching which cannot be ruled. Paternal lineage cannot be distinguished clearly. The another disadvantage, it is exactly very costly. Around 400 American dollar is required for this test. Now, it is exactly the summary of the total fetal DNA determination and sex determination of male and female babies, exactly followed by only the different loci in Y chromosome. Now, few short questions are there. As it is very effective after 7th week of gestation. Some say at 20th week of gestation, it is 99% effective. But it is ineffective before 5th week of gestation. Now, during this estimation, the fetal DNA and the maternal DNA, which are collectively found in the serum or plasma. But how they can be distinguished? The fetal DNA having the short length end of only around 200 base pairs, and 11 to 13% of the total free DNA are found, whereas maternal DNA are long in length, having the more base pair around 500 and more, and 80 to 90% of the total free DNA are found as the maternal DNA. Now, another point to note, after after 2 hours of birth, this type of DNA are not found at all in the maternal blood or plasma. So it is a very sensitive test. Now, the another important point to note that the cell-free DNA, the cell-free DNA not only is used for this diagnosis, it is also used in latest by different anomalies and different diagnosis tests for the pathological conditions. Now, if you have any questions on this, you may ask now by WhatsApp, by phone, or by mail. Thank you.